Hi everyone and welcome to our video on some exam questions related to biological molecules. In specific, in today's video, we are going to be looking at proteins and enzymes. Our first question is from the 2016 AQA AS Biology Paper 1 and states, describe the induced fit model of enzyme action. So this isn't really an application question, it's something that you should know, and the technique comes with exam practice of what the mock scheme actually wants, which is what we are going to be having a look at in today's video. So the induced fit model of enzyme action is the model which has newly been introduced. At GCSE, you would have looked at the lock and key model. At A level, we've been introduced to the induced fit model. And the idea of the induced fit model is that the active site of the enzyme is not complementary in shape to the substrate. And therefore, in order for the substrate to bind to the active site must slightly change shape. And when it changes shape, remember enzymes are a type of protein, and a protein contains different sort of types of bonds, disulfide, ionic, and hydrogen bonds. When the active site of an enzyme changes shape, it puts stress on the bonds. And that's basically the induced fit model of the enzyme. If I show you the mark scheme for this question, it says active site is not complementary. You need to state that. That's a key sort of word, complementary. It's not complementary to the substrate. Therefore, in order for the substrate to bind to the active site of the enzyme, the shape of the active site must change shape in order for that enzyme substrate complex to form. And therefore, the active site changes, and because it changes, it puts stress on the bonds. Okay, and that would get you 2 marks. Okay, you only need 2 of these points in order to get 2 marks. Okay, moving on to our next question. It says, a scientist investigated the hydrolysis of starch. He added amylase to a suspension of starch and measured the concentration of maltose in the reaction mixture at regular intervals. His results are shown in figure 4. Explain the results shown in figure 4. So in figure 4, we're given a graph. We're given time on the x-axis in minutes, and we're given the concentration of maltose on the y-axis, and we can see that there's a curve. Okay, and we just need to explain these results. So what we can conclude from this graph is that as the time increases, the concentration of maltose produced also increases, but at a decreasing rate, because the line starts to level off, and that's what we need to describe. Okay, now remember, amylase is added to a suspension of starch. Okay, so starch is present and amylase is added. Amylase is the enzyme. Now, why would it level off? Okay, so as the rate increases, the amount of starch being used up also increases. Now it slows down, the concentration of maltose produced slows down because the starch is being used up. Okay, and we can see that after 25 minutes, which is at this point here, we see a very minimal increase in the concentration of maltose. That tells us that there is either none or very little amount of starch left in the suspension. Okay, and that's what we need to explain. Okay, so first explain the general sort of trend of that line, and you can say that the rate of increase of concentration slows down through as the time increases because the starch is being used up. Okay, so the rate of increase in the concentration of maltose produced slows down. And why is this? Is because the starch is being used up. Because the starch is being used up. Now you will need to zoom in into a specific point and explain that point to get the second mark. And the easiest place to do it would be this place. Okay, after 25 minutes there is very little starch left. Okay, that's why it sort of levels off. Okay, so the line levels off after 25 minutes as very little starch left. Okay, and that's what I would say in my answer. If we have a look at the mark scheme, okay, I'll just get rid of my writing. If we have a look at the mark scheme, it says increase in the concentration of maltose slows as substrate or starch is used up. Or you could have said that there is a high initial rate as plenty of starch or more enzyme substrate complexes can be formed. So we focused on this part of the graph. You could have also focused on this part of the graph and said that initially the rate of concentration of maltose being produced was really high as there was enough starch so more enzyme substrate complexes could be formed. Okay. However, the second mark is looking at this specific point after 25 minutes. It levels off because there's no starch left. And it does say except little. Okay? And that's what you need to write for 2 marks. Next question. A quantitative Benedict's test produces a color whose intensity depends on the concentration of reducing sugar in a solution. A colorimeter can be used to measure the intensity of this color. The scientists use quantitative Benedict's test to produce a calibration curve of a colorimeter reading against concentration of maltose. Describe how the scientists would have produced the calibration curve and used it to obtain the results in figure 4. So now this isn't something that you are taught, it's more of the practical skills that you need to apply, and it's how you would produce a calibration curve. And again, it's a common exam question, so with exam practice you will get to know this exam technique. Okay, how to produce a calibration curve. Well, basically you need to produce your maltose solutions. Okay, whether unknown or known concentrations, you need to produce your maltose solutions, and you need to use a colorimeter to measure the colorimeter value, the color value of each solution. And then for each of those maltose solutions, you need to plot the colorimeter reading on a graph. Okay, and then you find the concentration of the sample that you're looking for after you've drawn your calibration curve. So if I show you the mark scheme for this question, so you produce your maltose solutions with known or different concentrations, and you carry out a quantitative Benedict's test on each. That's in brackets, so you don't really need that. What they're looking for is that you produce your maltose solutions. The second mark comes from saying that you measure the colorimeter value for each solution and plot the calibration curve or graph. And then you can find the concentration of sample from the calibration curve at a set value. Okay, now if you do mention the axes, which many people tend to do, they must be mentioned correctly. So on the x-axis, so going across, you have to have concentration, and on the y-axis you have your colorimeter reading. I'll put color reading for sure. Okay, so just remember that if you mention your axis, they must be correct or you lose your second mark. So again, this sort of method is the same for nearly every exam question that comes up on the calibration curve for 3 marks. So I would just recommend learning these 3 points. You produce your concentration of solutions, you measure the calorimeter value of each solution and plot a calibration curve, and then you find the concentration of the sample of interest from the calibration curve. Okay, and that would be worth 3 marks. Our next question is still from the 2016 AQA Biology Paper 1, and we're given a passage. Now, I'm not going to read through the passage, I'm just going to go through the questions and refer back to the passage when necessary. So, the first question says, suggest how amyloid precursor protein can be the substrate of 2 different enzymes, alpha secretase and beta secretase, lines 3 to 5. So, the lines of interest are these 3 lines here. Now, if we just have a look at that statement, we are given 2 enzymes, alpha secretase and beta secretase, and we're given one substrate, and it's saying that that substrate, it's complementary in shape to the active site of both enzymes. How would that be possible? Because each substrate is specific to each active site. And the only logical explanation that you can have here is that each enzyme, we've got these 2 enzymes, we've got alpha secretase and beta secretase. Each of these enzymes will be complementary to the substrate, but will be complementary at a different part to the substrate. Okay, it will be complementary to a different part of that protein. Okay, so this protein, amyloid precursor, is the substrate to 2 enzymes. That means that this protein, if we were to draw it out, we could have something like this and something like this. Okay, and one enzyme could be like this and one enzyme could be like this. Okay, as you can see, they're different shapes. One's curved, this is one enzyme, this is another enzyme, and this is the same substrate. Okay, and as you can see, both enzymes are binding to the same substrate. So basically what this question is trying to get from you is that each of those enzymes, they are complementary to a different part of the protein substrate. If I show you the mark scheme, different parts, areas / amino acid sequences of protein, each enzyme is specific or complementary to a different part of the protein. Okay, and if you write this second point, you will get both marks. Okay, so what this is getting at is just that the 2 enzymes are specific to the same substrate as they have bound to different places on the substrate. Now, the second part of this question says one product of the reaction catalyzed by beta secretase is a smaller protein. Describe what happens in the hydrolysis reaction that produces the smaller protein from amyloid precursor protein. Now, this is basically a knowledge question and it's not really an application question. The answer to this is clearly mentioned on the spec. Okay, because what happens during a hydrolysis reaction and producing a smaller protein? You're basically breaking up a larger protein which has many amino acids bound to it, and you're adding water because it's a hydrolysis reaction. Condensation reactions, they release water. Hydrolysis reactions, they use water. And when talking about proteins and amino acids, what bond are we talking about? We are talking about the peptide bond. Therefore, those are your 2 marks. Peptide bond is the bond which is broken by using water. Okay, and the mark scheme is really simple there. Peptide bond is broken by using water. And that would be worth your 2 marks. Okay, final set of questions. Same passage, It says many people with Alzheimer's disease have mutations that decrease alpha secretase production or increase beta secretase production. Lines 8 to 9. Use the information provided to explain how these mutations can lead to Alzheimer's disease. And it's worth 3 marks. So if we go to lines 8 to 9, we're looking at basically these two lines here. And it says many people with Alzheimer's disease have mutations that decrease alpha secretase production or increase beta secretase production. Basically what we're given in the question. Now, it says explain how these mutations can lead to Alzheimer's disease. Well, if we read just before that, it says one product of the reaction catalyzed by beta secretase is a smaller protein that can lead to beta amyloid protein formation. Okay, now what we're looking for here is that the alpha secretase production is the enzyme of interest. Okay, the alpha secretase production is the functional enzyme. Okay, and by having a mutation, it's decreasing the production of the functional enzyme. Okay, because it's decreasing the production of that functional enzyme, that means that there will be more substrate which is left to bind to the beta secretase enzyme. And if beta secretase enzymes have more substrate bound to it, that will cause more plaque to be formed, as we can see through this text. Okay, it says here this involves small beta amyloid proteins binding together to form structures called plaques in the brain. Okay, so if we increase the amount of substrate that can bind to beta secretase, that means that there will be greater plaques which are formed in the brain. And that's the effect of the mutations and how it can lead to Alzheimer's disease. So if you did have a read of this text, Alzheimer's disease leads to dementia, and that involves small beta amyloid proteins binding together to form structures called plaques in brains. Beta secretase is basically causing those beta amyloid proteins to be formed. Okay, and with a reduction of alpha secretase enzymes, that's causing more substrates to bind to the beta secretase enzyme, and therefore more substrate, i.e. beta amyloid proteins, are forming, and that's what's causing the plaques in the brain to form, which is leading to dementia. Okay, so if I show you the mark scheme for this question, it says mutations prevent the production of the functional enzyme. If you increase the beta secretase enzymes, as it says over here, there's an increase in beta secretase production, that's going to lead to more beta amyloid production. And if there's more beta amyloid production, that's going to lead to greater plaque formation. And that's what leads to Alzheimer's disease, which then leads on to dementia. Okay, so through this passage, it's just about making those connections. And that's where the 3 marks are coming. Normally you would think, oh, I can't find the answer. Well, the answer is not in the text. You have to make connections with the different part of the text to get a reasoning for each point you make. And that's where the 3 marks are coming from. Okay, the next question says one possible type of drug for treating Alzheimer's disease is a competitive inhibitor of beta secretase. Explain how this type of drug could prevent Alzheimer's disease becoming worse. And it's worth 2 marks. Now, if we have a look at this question, it's more of a knowledge question. Once again, it's not really an application question, because what does a competitive inhibitor do? A competitive inhibitor will bind to the enzyme's active site, preventing the substrate from binding to the enzyme, and therefore you will get less product which is formed. And that's basically what this question is asking for. However, the only difference is that you need to apply it to the scenario you're given of Alzheimer's disease. Okay, so basically this competitive inhibitor is going to bind to the active site of beta secretase. Okay, it's going to bind to the active site of beta secretase, and it's going to reduce the production of your beta amyloid protein formation. And that's what the mock scheme says. Binds to the beta secretase enzymes and reduces the production of beta amyloid. Okay, or you could have said plaque. Final question. When some of these types of drugs were trialed on patients, the trials were stopped because some patients developed serious side effects. Using the information provided suggests why some patients developed serious side effects. Now, we're given that information in this last paragraph, okay, but it doesn't really give us why they developed those serious side effects, or it doesn't even give us any hint as to why they develop those side effects. So this is a fully application question, and you will have to think to yourself, well, what's happened? We've added a competitive inhibitor, and that's prevented the amount of B amyloid production. Okay, that's prevented the B secretase from binding to the substrate to produce B amyloid. Okay, so that may be the problem. That's what this question is getting at, because once the competitive inhibitor has been added, it's caused side effects. That means the beta amyloid protein does have some sort of positive effect. Okay, and the answer for this question is basically that you need some of the beta amyloid to prevent the side effects. So the side effects that occur, you still need a small amount of beta amyloid in order for those side effects not to occur. However, this competitive inhibitor is inhibiting it sufficiently enough to prevent enough beta amyloid from being produced. The mock scheme says some beta amyloid required or needed to prevent the side effects, or some beta secretase is needed, or you could have also had leads to buildup of amyloid precursor protein that causes harm, or too much product of alpha secretase which causes harm. Okay, so anything like that. But this is a fully application question, so you need to judge the scenario you're in before you write down your points. And that is it for this video. Thanks for watching. I hope you liked it. And one last thing, please subscribe, hit the like button and the notification bell.